All right, everyone. Welcome to the first lecture of the last unit of Bio 139. We are in the last little bit of the semester, so this chapter is really going to kind of tie everything together. It puts a nice little wrapping and bow on everything we've talked about this semester. It also really pulls together a lot of Bio 137 with what we've learned this semester. So even though this chapter comes before everything we've done so far in our textbook, it really does work well as a way to kind of pull everything together at the end, which is why we cover it last. So, as always, let's start with our not really attendance questions. What are the two major communication networks in the body? And what are the big differences in the two? So these may require you actually to go back and review some Bio 137 if they are not immediately popping into your head. So go ahead and pause the video and try to answer these and see what you come up with. And let's see what the answers are. So the two major communication networks in the body are, of course, the nervous system, covered in Bio 137, and the endocrine system, mentioned in Bio 137, and what we're about to cover now. And what are the big differences in the two? This was probably discussed in your Bio 137 class. Well, the big differences are the mechanisms with which they work, the speed at which they work, and the duration of their effects. So let's see what that means. The nervous system communicates using a combination of electrical signals and neurotransmitters, while the endocrine system is going to use hormones. The nervous system has really quick response time. When a neuron sends a signal, there's a very immediate response. And the endocrine system, there's actually quite a delay. The endocrine system is pretty slow to respond in most cases. But in the nervous system, even though there's a really quick response, that response is over just as quickly as it began. But on the endocrine side, the response is very long-lasting. So once a hormone has an effect, that effect remains in place for quite a while. Just a couple of housekeeping items before we dive into the material for this lecture. This chapter, not so much this lecture, but the remaining lectures, this chapter and the chart in Lab 12 go hand in hand. Even though lab and lecture have really tied together nicely this entire semester, even if the pacing was a little off now and then, this chapter and the last portion of lab, we can really think of it as one big class. The things that we're going to need to know for each are almost identical. They overlap so much, we can just think of lecture and lab from this point as one single thing. It's all the same material. We're going to be covering the same hormones, and in most cases, the same details of those hormones. So when we look at the outline for this chapter, I've actually taken the liberty of filling out the vast majority of it for you. Now, I do expect you to add quite a bit to it, but the majority of it I've already filled out for you. And that can really help you fill out that lab chart. Now, that doesn't mean just take exactly what I wrote and write it on that lab chart, because that won't do you any good. But the details that I provide for you are the foundation for what you'll put in that lab chart. And when you get to lab exam 3, that lab chart is going to be something you really want to understand. Now, memorization will only get you so far. You actually have to understand what's there. And that's the point of these next several lectures after this one. I'm going to explain all of that material to you. But if you know and understand that lab chart, then on lab exam 3, you're going to be able to understand and answer most of the questions. Because when you see the models, when you see the slides, when you see the questions on the just like the regular non-model and slide questions, a lot of that material is going to come from that chart that's in Lab 12. And that chart that's in Lab 12 is what we're going to be covering pretty much in these next few lectures. So when you look at a picture of a model, well, you may see something like, what is the name of this gland? But you may also see, what is a hormone released from this gland? Or what is an oversecretion or a hypersecretion associated with this gland? Or what is a stimulus for the hormone released from this gland? And all of that comes from the chart. Now, also, don't forget, I know we're at the end of the semester and hopefully you've been looking at these all along, but my Facebook and my YouTube. My Facebook has a lot of images that are not in the lab manual. And my YouTube channel, which I believe Dr. Weitzgarver posts, and I know I post, depending on who you have for lab, you will be able to see a lot of these things in the videos that I have on my YouTube channel that will really help you for lab. So if you have an instructor that does not post my videos, they can all be found on my YouTube channel. So with that out of the way, let's start talking about the endocrine system. And this lecture is really just going to lay kind of the foundation. It's going to introduce some concepts that we're going to be talking about. It's going to talk about hormone basics, but then the remaining lectures are going to actually be talking about the specific hormones and the glands. So let's learn some definitions that we're going to be using. And we've used these terms off and on all semester, really even as far back as Bio 137. But something that I've learned is a lot of times, even this late in Bio 139, students might not really fully understand these terms. So let's give some definitions. First, stimulus, or the plural stimuli. A stimulus is something that is going to cause the release of a hormone. In general, a stimulus is just something that causes a response. But in this chapter, since we're talking about hormones, a stimulus is what causes a hormone to be released. Next, function. In this chapter, we're going to be asking, what do the hormones do specifically? So for ADH, we could say ADH raises your blood pressure, or we could say ADH causes water reabsorption or something like that. But we could be even more specific. How does it do that? And that's the mechanism of action. How does a hormone cause what it causes? So keeping with ADH, ADH causes vasoconstriction. It's a vasoconstrictor. Remember, its other name is vasopressin. So it causes smooth muscle to contract in the blood vessels. As far as water reabsorption, it causes the collecting duct, and really also the late DCT, but not so much important there. It causes the collecting duct to insert aquaporin type 2. So those are the mechanisms of action. Stimulus, what is going to cause the hormone to be released. Function, what does the hormone do exactly? Mechanisms of action, how does the hormone do what it does? And then we will also be talking about inhibition. And inhibition is something that either blocks the release of a hormone or blocks the effects of a hormone. So inhibition, something that blocks a hormone from doing whatever it does. So let's talk about some differences in endocrine glands, which is what we're going to be focusing on in this chapter, and exocrine glands, which we've talked about a few times here and there throughout the last 2 semesters. First, what is an exocrine gland? An exocrine gland we see here in image A. Exocrine glands, first, they have a duct. Exocrine glands have a duct that they release their product through. And that duct either empties out onto the surface of the body. Think about sweat glands. Sweat glands are exocrine glands. They release through a duct onto the surface of your skin. Or they also can release into a hollow part of your body. For example, some of the glands in your stomach and small intestines, or in your pancreas, they release into a duct, and then that duct releases into the digestive tract, a hollow place. Endocrine glands, which we see over here in B, are ductless glands. They do not have a duct. They secrete their product, hormones, directly into the bloodstream at a capillary. So exocrine have a duct, and they secrete their product through the duct into either a hollow portion of the body or onto the surface of the body. Endocrine glands are ductless glands, and they secrete their product directly into the bloodstream at a capillary. Well, some other terms we might hear are autocrine glands, paracrine glands, or autocrine hormones, paracrine hormones more specifically. So typically when we talk about an endocrine gland and a hormone, those hormones will enter the bloodstream and often have effects in completely different parts of the body. For example, ADH. ADH is released by a structure in the brain, and it travels in one place to the kidneys. Well, autocrine hormones are released by a cell, and they stimulate the same cell that released it. Oftentimes it's a positive feedback. Not always, but oftentimes it's a positive feedback. But it is released by the same cell that it then in turn stimulates. Paracrine hormones, very, very similar. Paracrine hormones are released by a cell, and then it stimulates nearby cells, neighboring cells. So autocrine and paracrine are very, very similar. Autocrine stimulates itself, paracrine stimulates its neighbors. And even though our book doesn't really talk about it much, and it's not really a big point, I want to still go ahead and talk about pheromones. Pheromones are similar to hormones, but pheromones are released by one organism, and they stimulate another organism, usually another organism of the same species, but stimulates another organism than the one that released it. The perfect example of this is a lot of different insects. Moths, for example. A moth will release a hormone when it's ready to mate, and it will attract another moth of the same species so that that moth knows where the moth is and that it's ready to mate so that they can find each other and procreate. Now, whether or not humans have pheromonal activity, it's kind of the subject of a debate. A lot of evidence says that yes, as a matter of fact of all things, body odor seems to have some pheromonal activity, but not important for this chapter. So let's talk about classes of hormones. There are different types of hormones that behave differently based on their structure. So first, let's talk about steroid hormones. Steroid hormones tend to end in OL or ONE. So estradiol or aldosterone or testosterone or cortisol, something like that. Now, all steroid hormones are based on cholesterol, and cholesterol itself is a steroid hormone. So there's cholesterol. Cholesterol is then going to be modified. It's going to have something added to it or something taken away from it so that it then becomes whatever hormone we're talking about. For example, progesterone or cortisol or aldosterone or testosterone or estradiol. So we can see all of these are derivatives of cholesterol. And all steroid hormones will behave the same way. That's what we see in the picture over here. Here we have just kind of our generic steroid hormone. Steroid hormones do not need a receptor on the surface of the plasma membrane of the cell that it's going to stimulate. Steroid hormones are nonpolar because they're based on cholesterol, and nonpolar structures are able to easily diffuse through the cell membrane. So steroid hormones pass through the cell membrane. Their receptor is right at the nuclear envelope. And this allows the steroid hormone to enter the nuclear envelope, and once it's bound to that receptor, it attaches to a gene and causes direct gene transcription. The steroid hormone binds to the receptor, which then binds to the DNA at a gene, and it causes transcription of that gene. So we get protein synthesis. Keep that in mind. We're going to come back to it in just a few moments. Another type is a peptide hormone. And you'll see when we go through all of the different hormones throughout the next few lectures, I group these together. So you do not need to know the different types of peptide hormones. I group them together, but peptide hormones are all based on amino acids. But they could be a single amino acid. They could be a peptide, which is a few amino acids stuck together. It could be an entire full protein, which is a bunch of amino acids with a very specific shape. I call all of those either peptide hormones or amino acid-based hormones. Now, with peptide hormones or amino acid-based hormones, these are polar. So they are unable to pass through the cell membrane the way those steroid hormones did, which means that we need a receptor on the plasma membrane of the cell that this is going to stimulate. If there is no receptor on that cell that is specific to the hormone, then that hormone will not be able to communicate with that cell. So what do we mean by communicate? Well, there's actually a few different ways of communication. There's something called the cyclic AMP or the CAMP second messenger system, which is what we're going to talk about here. There's also something called the DAG second messenger system or the IP3 second messenger system. Those are really kind of the same thing, DAG, IP3, second messenger system. We're not going to talk about those. They function very, very similar to what we're going to talk about here. So, little pink guys up here, this is an amino acid-based hormone. And big purple guy right here, this is the receptor that is specific to the pink hormone. Now remember, the way proteins work, they have a very specific shape. So this receptor is shaped in such a way that only this pink round hormone can attach to it and have an effect. when the hormone attaches to it and has an effect, now we're going to see some sort of cellular response. So let's look to see how the cyclic AMP second messenger system works. At rest, when this cell is not being stimulated, the receptor has another protein stuck to it called a G protein. And the G protein is just kind of sitting there, not really doing much of anything at first. But when the hormone attaches, it's going to cause a shape change. Remember, anytime something attaches to or detaches from a protein, it causes a shape change. So when this hormone attaches to the receptor, it causes it to change shape. And it causes this G protein to break free of the receptor. And now when it breaks free from the receptor, GTP attaches to it. GTP is very similar to ATP. It's, remember when we learned the different nucleotides of DNA and RNA? We had A's and C's and T's and G's and U's. Well, ATP was adenosine triphosphate. ATP was adenosine triphosphate. ADP was adenosine diphosphate. And GTP is guanine triphosphate. So GTP attaches. And now this G protein is going to travel to a nearby protein and attach to it. This nearby protein, also on the surface of this same cell, it makes it look like it's pretty far apart in this picture, but it's not. They're very close to each other. When this G protein with the GTP attaches to this other protein called adenylate cyclase, it's going to cause the adenylate cyclase to take the ATP that's in this cell and convert it to something called cyclic AMP. Well, remember, ATP is just a phosphate, an adenosine with 3 phosphates attached to it. If we took a phosphate off, it became ADP. If we took another phosphate off, it became AMP. Well, that's what we've got right here. All we did was take 2 of the phosphates off. It went from ATP to AMP. Cyclic AMP is now going to find a protein kinase, which is just a protein that's going to cause something to happen. And there's lots of different protein kinases. But this inactive protein kinase is activated by the cyclic AMP. It becomes an active protein kinase. And really, oftentimes, this is going to then activate another, which then activates another, which then activates another. It's going to cause this domino effect. And at the end of the domino effect, we have some sort of activity within the cell. Lots of kind of craziness in this picture, but really, if we back up and look at the big picture from far off, the hormone attached to a receptor on the surface of the cell, there was a domino effect that resulted in some sort of activity within the cell. That's really what I want you to know. What we can see, let's go ahead and compare the differences in the two hormones now that we've looked at them. Steroid hormones went straight to the nucleus of the cell, had a receptor there, it bound to a gene, it caused transcription, so we synthesized proteins, and then we had whatever effect we were going to have. Peptide or amino acid-based hormones instead must attach to a receptor on the surface of the cell, which then causes this chain reaction, which then has some sort of effect within the cell. So I do expect you to need to know the differences. You do not need to know all of the gritty details on that second messenger cyclic AMP. But let's kind of, while we're on this slide, ask ourselves kind of a mini thinking cap question. Why would we want two different types of hormones that behave so differently? What would be the benefit of having two different types of hormones? So I'll let you think about that. I ask that you pause the video, treat this just like a thinking cap question, before listening to what the answer is. Well, steroid hormones, since they can go directly through and cause activation of a gene, they are indiscriminate. They will have effects on every cell in your body. So they have very widespread effects that tend to just be, they increase metabolism. But peptide hormones, they can only affect cells that have a receptor for them. Why would that be at all beneficial? Because it allows us to be very, very targeted. It allows us to target specific cells. So think, why does ADH not have an effect on bone cells? Because bone cells don't have a receptor. There's no need for ADH to target bone cells, so those bone cells don't have a receptor. ADH can target smooth muscle within the blood vessels, and ADH can target those cells late in the nephron because those cells have receptors. Whereas something like testosterone can target lots of cells throughout our body because it doesn't need a receptor. But there actually is another type called biogenic amines. And biogenic amines kind of behave like both steroid hormones and peptide hormones, or amino acid-based hormones. And the two that we're really going to talk about in this chapter are thyroid hormone, thyroxine, T4, and epinephrine. Well, thyroxine from the thyroid, it really behaves more like a steroid hormone. Even though it's amino acid based, it behaves like a steroid hormone. It has very widespread effects, and even though it's an amino acid based hormone, it behaves like a steroid hormone. Epinephrine, on the other hand, behaves more like an amino acid based hormone. So they kind of have properties of both. Okay, so now let's change gears and talk about stimuli, things that cause hormones to be released. And there are different classes of stimuli. First, let's talk about humoral stimuli. Humoral stimuli are going to respond to levels of something in the blood. Remember way back when we were talking about the immune system, when we talked about humoral immunity, we said that humors just meant the liquids of the body. It's an old term. Well, humoral stimulus, it's something in the liquids of your body, specifically blood, that's going to cause a hormone to be released. In the picture that we have here, we see humoral stimulus, calcitonin, or parathyroid hormone. Calcitonin and parathyroid hormone respond to levels of blood calcium. This picture showing us parathyroid hormone. Calcitonin from almost the same area behaves almost the same way, but we are monitoring levels of calcium in the blood, and when calcium is too high, calcitonin is released. When calcium in the blood is too low, parathyroid hormone is released. So, levels of something in the blood causing a hormone to be released is a humoral stimulus. Next, neural stimuli. This is when a neuron or neural input is going to cause the release of a hormone. Think back to Bio 137 when we talked about the sympathetic nervous system. When the sympathetic nervous system is activated, we get very quick effects from norepinephrine from our neurons. But at the same time, some of those neurons actually stimulate the adrenal medulla, the middle of the adrenal gland, to release epinephrine and norepinephrine into the blood. And this then has a very long-lasting effect. If anyone's ever jumped out and scared you, you get that very immediate response of, oh God, I'm scared, I'm going to either fight or pass out or something. Well, that's because of the nervous system side of your fight or flight response. And then, even 5 minutes later, you still got that kind of tense, jittery feeling. That's because of the endocrine side of your fight or flight response. So in this case, neural stimuli from the nerves caused the release of the hormones into the blood. That's a neural stimulus. And the third type is hormonal stimulus. I'm going to write another word here, tropic. When a hormone causes the release of another hormone, it is a tropic hormone or it is a hormonal stimulus. But I don't believe your book really talks about this, but it is very important. In some cases, a hormone may stop another hormone from being released. That is also a hormonal or tropic hormone, a hormonal stimulus or a tropic hormone. So if a hormone causes another hormone to be released, or if a hormone causes another hormone to stop being released, that is hormonal stimulus. Now, our next lecture, when we're talking about the pituitary gland, I don't know if it's the next lecture or the one after, but when we talk about the anterior pituitary gland, we're going to be talking a lot about hormonal stimuli. All right, so the hormones get released. What about transport? How do those hormones get transported about the body? Well, that's going to depend on what type of hormone it is. Amino acid-based hormones were polar. Steroid hormones based on cholesterol were nonpolar. So since the blood is mostly water, there's going to be a distinct difference in how these are going to be transported. Amino acid-based hormones, polar, well, they dissolve well in water. So when a cell releases a polar hormone, it can just travel through the blood as is. But steroids are nonpolar, or think back to Bio 137, here's a word for you, hydrophobic, water-fearing. They do not dissolve well in water. Well, think about if you've ever put oil in water, what happens? Well, cholesterol is a fat. If you put it in water, it's not going to mix well. So we can't just put steroid hormones into the blood. They would not dissolve, they would not mix, and that would cause all kinds of problems. Instead, most of them get attached to a carrier protein, something like alpha and beta globulins. Tying it all together, we talked about alpha globulins and beta globulins on the first day of class. Now we're seeing, what are they transporting? Well, one of the things they transport are steroid hormones. There are some steroid hormones that do not get bound, but the vast majority are bound to those transport proteins. Well, what about modulation of sensitivity at the target cell? All cells do not respond to their hormone exactly the same. There's something called upregulation, in which the hormone causes the target cell to make more receptors for that hormone, which then allows that hormone to have more of an effect because that cell is more sensitive. So here in this first picture, this cell has a few receptors to the hormone. But one of the things that this hormone does when it attaches is it causes the cell to make more of those receptors. Now when that hormone is released, this cell is going to have a bigger response because it's got more receptors for that hormone. The hormone caused the cell to be more sensitive to that very hormone, so that hormone could have more of an effect. That is upregulation. Downregulation is exactly the opposite. A cell that has a lot of receptors, when the hormone attaches, it causes that cell to get rid of some of those receptors. Well, now the cell has fewer receptors, so it's going to now have less response. Well, how do different hormones interact with each other? Well, it depends on the hormones. First, there's something called synergistic effects. And this is when some hormones are released at the same time, they magnify each other's effects. For example, FSH causes testosterone to have a bigger effect than it would otherwise. FSH causes testosterone to have a bigger effect than it would otherwise. But at the same time, testosterone causes FSH to have a bigger effect. So FSH and testosterone are synergistic. They cause each other to have a bigger effect than they would otherwise. Next, permissive effects. And this is when one hormone is released earlier, it causes the target cell to become more sensitive to a hormone that is going to be released later. And a good example of this is estrogen and progesterone. When estrogen is released, it's going to bind to some cells, and it's going to cause those cells to make more progesterone receptors. That way, when progesterone is released later in the cycle, those cells have more receptors for it, so they are going to have more response to the progesterone. But interestingly, until estrogen is released, those cells will not respond to progesterone. So estrogen not only is permissive for progesterone, it's required for progesterone effect. And lastly, antagonistic effects. And this is when a hormone is going to block the effect or block the release of others. For example, estrogen, again with estrogen blocks the release. It doesn't block the release. It blocks the response to prolactin. So when prolactin is being released, mammary glands would respond to it, but estrogen blocks those mammary glands from responding to prolactin during pregnancy. There's no need for prolactin during pregnancy in most cases. So until the pregnancy is nearing its end, estrogen is prohibiting or blocking the response to prolactin. When we get near the end of the pregnancy, the estrogen is going to kind of stop, and then the prolactin will cause the mammary glands to start to produce milk. Also, insulin and glucagon. Insulin, which is insulin and glucagon both we're going to study at the end of this chapter, but insulin is going to lower your blood sugar. Glucagon is going to raise your blood sugar. But they're not released at the same time. If they are being released at the same time, they are antagonistic to each other. And once a hormone has its effects, we need to get rid of those hormones and stop their effects. And that is hormone clearance. And different organs are going to work with hormone clearance. For example, the liver. Liver is going to metabolize various hormones. It's going to break them down so that they no longer can have an effect. Kidneys will remove the hormones from the blood and they will leave in the urine. Target cells that are responding to the hormones are going to cause lower levels of the hormones in the blood. So after a hormone has had its effect, we don't want it to stay there so that it can keep having an effect. It needs to be stopped, and that's hormone clearance. All right, so that is the end of this first lecture, when all we've done is really just talk about the basics of hormones. Our next several lectures are going to start looking at specific glands and the hormones that they produce. All right, take care, and I will talk to you next time.